Good afternoon, everyone. My name is Hongli. I'm from Fungal Genomics Evolution Biological Research Center. Today, I'm going to talk about investigation of gene regulatory networks underlying pattern formation in coprinopsis cinerea. Complex multicellular organisms are dominating the visible world. They are subjected to intense research, and the development of morphological patterns of multicellular organisms has been well elucidated in plants and animals. In animals such as Hox genes, which were identified in Drosophila in the first time, were found to be able to specify regions of the body plan of an embryo along the head-tail axis of animals. And the PAX genes is another gene which were found to be able to control the development of eyes and other sensory organs. What's more, dissimilar organs such as eyes from different animals long thought to have evolved separately are controlled by this similar gene toolkit. And in plants, the ABC model of flower development is also well elucidated and widely studied. Gene networks were also investigated in model plant systems. It showed that flowering plants produce a pattern of gene expression in meristems that leads to the appearance of an organ oriented towards sexual reproducing, which is a flower. And fungi are also morphologically diverse and can form spectacular pattern, but the most fascinating structure among fungi is mushroom, and among mushroom, the most complex mushroom morphology is a pileate stipated type. Mushroom development is a highly compelling area of research, deserving thorough investigation not only due to the existing knowledge gap, but also owing to its significant potential in the industries. For example, mushroom cultivation is a large global industry. Now and soon, it can reach a bigger market in 2030. And also we have already had good examples of applying the development-related research to industry. One is the sporeless oyster mushroom, which can significantly decrease the environmental impact of spores and also reduce the respiratory issues of workers. Another example is the common white button mushroom, which we like to eat, has been modified to resist the browning. So studying the pattern formation of mushrooms has the potential to bridge gaps in our understanding of mushroom development and lead to valuable applications in the mushroom industry. Previous knowledge accumulated in mushroom development reveals that pattern formation in mushrooms occurs in the early primordial stage, and in a very early time, histological illustrations of a coprinoid species have already suggested how cell differentiation and early pattern forms from the noodles to the secondary initiation stage, and then especially from the third image, it's easy to tell that a generative hyphae grown out from the top of the noodles and formed a pileate rudiment. So in our study, we are using pileate stipated forms. as a model for understanding pattern formation in fungi. Pileated stipitate mushroom has a well-defined cap and a stipe. And here in our study, we are more interested in cap development in pileated stipitate type mushrooms to identify genes underlying cap development. And firstly, we focused on transferrin factors because in mushrooms, not so many genes were well annotated and functionally studied. CRISPR-Cas9 system is not working very efficiently. So starting with transcript factors, try to build gene regulatory networks of cap development could be a tipping point in pattern formation study in fungi. And this study includes whether our candidate transcript factors is a master regulator and what are their target genes. So to reach this point, we use Coprinopsis cinerea as a model species to examine cap development. With an increasing of knowledge of developmental transcriptomics, we collected transcript factor genes that showed typical expression peak in cap. And also, all agaricales share a single fruiting body forming ancestor, which suggests there should be a core deeply conserved genetic toolkit of fruiting body development. Therefore, we mainly focused on the conserved transcription factors. And the conservation of these transcription factors was analyzed based on 137 fungal species. So in addition to the sequence conservation of genes, there should be conserved gene expression pattern across pileated stipitate species. Since developmentally regulated genes that are relevant for the development of fruiting bodies in general from genes with species-specific developmental rules, we examined the expression patterns in another 4 species. And they all nicely showed a cap expression peak. And at this end, a CPR1 gene, which we named, popped up and showed both sequence and expression pattern conservation. Knocking out the CPR1 gene was performed using CRISPR-Cas9 system, and it abolished the cap development. In the meantime, it cooks healthy vegetative mycelia. And from the morphology of the fruiting body, we observed that the development of the universal view seems to be affected. Sections of the wild type and the mutant strain also showed both the cap were differentiated in the 2 primordial stages. And to examine whether the capless phenotype was indeed caused by CPR1 gene, we reintroduced it into the CPR1 mutant again, and it restored the cap development. And also it led to a mature fruiting body. These results clearly proved that our phenotype is caused because of CPR1 gene. So then we came to the next question. Is CPR1 a master regulator? So our hypothesis is that a master regulator should be able to initiate cap development when expressed outside its natural context. And we came up with the idea that ectopically expressing CPR1 and either a tissue-specific gene promoter or a head shock gene promoter, because in the first case, the CPR1 expression could be induced by a specific tissue, and in the second case, the CPR1 inducing can be controlled manually by heat shock. So expressing CPR1 driven by the step-specific gene promoter in CPL1 mutant also restored the cap development, but with a different shape and size of the cap. And also expressing CPL1 driven by the heat shock gene promoter in the mutant restored the cap development again after heat shock. And without heat shock, there is no cap development. So based on these experiments, we came to the conclusion that the cap shape and size varied with different CPL1 expression level. Like under the different promoters, the cap shape and the size can be very different. Interestingly, always there is only one cap initiated under whatever promoter and always on top. So we came to question that, is there a one cap initiation strategy that is a promoter independent mechanism for ensuring only one cap develops? And this question, we don't have answers yet. And with all these results, I would like to summarize. So we found the CPL1 transcript factor, which can abolish the cap development. And this gene, based on expression level, influences cap shape and size. In addition, in any case, we didn't observe a cap initiation on the mycelium. We are also curious about why there is always only one cap initiated. Finally, I would like to thanks for all our lab members from CRISPR team and the bioinformatics team. And thank you very much for your attention. I'm happy to answer questions.
Thank you for your great presentation and welcome to the mycology department.
Thank you for inviting me.
The audience of the mycology department has already learned a lot about the evolution of fungi and fruiting body development and genetic background, and your lecture took us deep into the genetics so thank you again. My first question, is this an ongoing project, or have you already finished it and published the paper?
Actually, it's ongoing project. We didn't publish.
Okay, then makes it exclusive, your presentation. You mentioned also another interesting term, ectopic expression. It is an out of context expression. Is it the natural phenomenon when a mushroom grows on top of another mushroom, for example?
Actually, this not happens very often, and it's more like a natural context if another cap grow from the mushroom. But we are expecting this kind of phenotype when we try to induce the cap gene from the step. We are expecting the cap can grow from the step.
Oh, I see.
Mm-hmm.
Your lab reported more than 900 conserved developmentally expressed gene families. How many of them went through this comprehensive functional testing like CPL1? Not the exact number, but percentage.
Actually, in our lab, in the recent published paper, we also summarized the published genes which have been identified and also developmentally regulated in other mushroom. But these are really small number. It's like less than 100 genes were identified. Yeah.
So there is a lot to do.
So actually, in our lab, our colleagues they are trying to identify more transcription factors or some metabolites related proteins. So in the future, we will publish more genes.
Okay, thank you, and please come back to mycology department to present your new results. Thank you again for your lecture, and for our audience, I thank for the kind attention. We will meet next month on second Wednesday. Thank you.
Thank you.